Artificial insemination has become one of the greatest discoveries of the last century. For the first time, this medical technology has been successfully applied in the UK in 1977, and it resulted in the birth of Louis Brown in 1978, the first human being conceived in a test tube. In the Soviet Union, this technology has given its first results in 1986. In 1990, on our planet, there were more than 20,000 of the so-called in vitro children, and by 2010, they can be counted in the millions. Application of this technology is not an idle curiosity or a passing fad. Unfortunately, according to the latest world statistics, every sixth couple is faced with the problem of infertility, and reproductive technology, for many of them, is the only solution to this issue. Until recently, out of all 100 performed in vitro fertilization, only 25 ended in the birth of children. Now, thanks to the promotion of reproductive genetics, this figure has doubled. The most important tool that we have, which we are now holding in our hands, is Preimplantation Genetic Screening. In simple words, it is an analysis that is done at the stage of 5-day-old embryo, and it makes it possible to transfer the embryo into the uterus. It helps to determine whether the child will be healthy. Before you begin the selection of worthy contenders for replanting, it is necessary to spend hard work over their cultivation. New technology, embryoscopy, incubator, which is considered the newest equipment in the technology of artificial insemination, helps the Latvian specialists in this clinic.
Чтобы вырастить эмбрионы до 5-го дня, самый лучший вариант их держать в эмбриоскопе.
To grow the embryo until the 5th day, the best option to keep them in embryoscopy, a hatchery where they are cultivated continuously from fertilization until the 5th day. And we do not have to choose them out to see how they are divided. Because in conventional hatcheries, we need to look at them every morning, at what stage they are. Embryoscopy helps to see all the stages of the embryo, how it is divided. And on the 5th day, when it is necessary to do a biopsy for genetic analysis, we can see when the right moment to make this a biopsy.
На 5-й день, когда надо делать уже биопсию для генетического анализа, мы видим, когда самый подходящий момент, чтобы сделать эту биопсию.
Если говорить о предимплантационной генетической диагностике, она начинается с того, что
If we talk about preimplantation genetic diagnosis, it starts with the fact that the embryologists do a biopsy of the embryo on day 5 and take a few cells of trafficted derm. At this stage of development, the embryo already has two different cell lines, the inner cell mass, which itself will develop into a child, the fetus, and trafficted derm, which will develop into the placenta. And we take it for analysis.
В плаценту. И ее мы забираем на анализ.
To understand all the processes that happen in the offices of Ludmila and her colleagues, molecular genetics, for a person not advanced in this area, it is immensely difficult, even with thorough explanations. More or less everything is clear at the very last stage, which Ludmila affectionately calls pictures.
Результат отображается следующим образом. По этой картинке дальше можно будет сказать, данный эмбрион можно переносить в полость матки или
The result is displayed as follows. Looking at this picture, we can say if the embryo can be transferred to the uterus or not. Say there is a couple. They have 4 embryos, so we see the embryo number 1. He, instead of 2 copies of the first chromosome, has 3 copies. The first chromosome is very big and carries a huge number of genes. Such embryos will never be able to stay in the uterus, so there is no need to inseminate it. It makes no sense. Here, for example, the so-called neutral members. Most of chromosomes are not correct. The embryo is of very, very poor quality, even by visual inspection. It may look normal.
Выглядеть вполне нормально и может быть тоже подсажен. То есть этот метод именно повышает возможность забеременеть с первого раза.
Modern genetics does not stand still. It finds new approaches to the most hidden parts of the human body. It gets more information about human genes and their mutations causing congenital abnormalities and infertility. Loud results of these approaches, the long-awaited and well, are pleasing to not only parents, but also their humble assistants in white coats.